Hello, my name is Dr. Mike Keller. I'm a professor and undergraduate coordinator in the School of Renewable Natural Resources, and I'd like to introduce today's ag issue. Conservation is a major issue that we face within natural resources, wildlife, forestry, and wetlands. Conservation has been an evolving issue in terms of where we do the conservation and what animals we focus. But what's always been true is conservation has involved populations of animals, the species of animals that live together in communities, their habitats, their physiology, and the topic we're going to focus in on today, conservation biology and conservation genetics. Now, without any further ado, I'm going to introduce Dr. Sabrina Taylor.
Hi, my name is Sabrina Taylor. I'm a professor in the School of Renewable Natural Resources. My research interests are in conservation biology and conservation genetics. Conservation genetics is a key process to biodiversity because it's needed to adapt to change, right? So if every individual is a genetic clone of other individuals, it means that things like a disease that affects one individual is going to affect all individuals. And so this is why we focus in on genetic variation. So we are looking at things like how much genetic variation exists on the landscape, how that's partitioned into structured populations, how much gene flow occurs among those populations, and what the levels of inbreeding are, because that has such a big impact on reproductive success and survival. So next I would like to introduce one of my PhD students, Alessandra Bresnan, who's looking at genetic variation in the captive tiger population in the US.
Hi, my name's Alessandra Bresnan, and I'm a third-year PhD student in Dr. Sabrina Taylor's Conservation Genetics Lab. So many animals that we love and care about are facing potential extinction. In fact, 97% of tigers have been lost in the past century in the wild. There used to be 9 different subspecies found in the wild. 3 have already gone extinct, and of the 6 remaining, 5 of them are found in captivity, and 1 is not even found in the wild, it's only found in captivity. The majority of these tigers are bred for commercial purposes, such as people going and petting the tigers, getting photos with these tigers, and these tigers are called generics. Generics basically have hybridized origins. So there's different tiger subspecies and they've been hybridized together, such as Sumatran tigers with Malayan tigers. And these are often highly inbred. Inbreeding is where related individuals are made to meet with one another, and it can actually lead to a bunch of health issues, such as being cross-eyed or bow-legged or having kinked tails. Some argue that these tigers actually lack any conservation value since they'd never be found in the wild as they are found here today. A small proportion of the tigers found here in the United States in captivity are managed through species survival plans, or SSPs, in zoos. In these species survival plans, the different subspecies are not allowed to hybridize and they're kept pure. And they're also managed in order to avoid inbreeding, as well as to make sure that they maximize genetic diversity. This is now being done with paper pedigrees, but thanks to advances in technology, we can now study the entire genome of an animal and use this data to then help improve the genetic management of the captive breeding of these populations. By including genetic data, we can improve the amount of knowledge we have based on these individuals and help use it to maximize genetic diversity in these captive populations as much as possible. So that way, if we ever need, we can tap into these captive populations as a resource for the wild tigers in the future. We can use genetic tools in order to study populations and monitor inbreeding levels. This becomes especially important for small populations because small populations often have high inbreeding levels. So these can be the populations that are found in the wild that are highly endangered, or captive populations that have a small number of founders. Founders are the individuals that are initially taken out of the wild in order to start captive breeding programs. We can also use genetics for other applications, such as identifying whether a species has different subspecies, or, as in the case of generic tigers, whether these different subspecies are hybridizing. With my research, I will be using whole genome sequencing in order to study the captive populations of tigers here in the United States. I want to use this genetic data in order to help the SSPs and the zoos improve their captive breeding programs and try to maximize genetic diversity as much as possible. You can do this by integrating the genomic data that's generated through whole genome sequencing. The work of zoos is so important because not only do they breed captive populations that can serve as a lifeline for endangered species, but they also invest so much into conservation efforts as well as research, such as my own. So, if you want to help out, you can try to support your local zoo, and that can work towards helping conserve the tiger as well as other endangered species. Another part of my research will be looking at the commercialized breeding of captive tigers, for certain practices such as cub petting. I'll be doing this by using whole genome sequencing on generic tigers in order to identify the different subspecies that are represented in these hybrids, as well as to find out their inbreeding levels. You can help by making sure that anytime you visit a facility that has tigers, do some research and make sure they don't do cub petting, because it fuels the commercialized breeding of these tigers, which in the end can actually hinder rather than help conservation efforts for the tiger.